Hello everyone, we are Group N and we are here to briefly talk about the chemical mechanism of Alzheimer's disease and introduce a very promising piece of research that was published only about a month ago. Now, as you might already know, Alzheimer's is a neurodegenerative disease that is caused not by virus or bacteria but by an amyloid. And an amyloid is basically a protein that is insoluble, misfolded version of proteins that are naturally present in our body. For instance, mad cow disease is caused by a type of amyloid called prion. Now, the particular amyloid that we would like to focus on is beta amyloid. This is the amyloid that causes Alzheimer's disease. It is processed from a protein called amyloid precursor protein, APP in short. APP is an integral membrane protein found mostly at synapses of neurons. So this cell membrane you're looking at here is the synaptic membrane, and APP breakdown is a two-step process. If you look at the left side, when alpha secretase is used, we get the normal product P3. But if beta secretase is used, beta amyloid is formed because, as you can see, beta secretase can't cleave APP as close to the membrane as alpha secretase. Now, beta amyloid starts as a single molecule, but they tend to bunch up. Now, the thing is, when beta amyloid is in cluster form, they can bind strongly to nerve cells receptors, forming amyloid plaque. So this brown cluster you see is the amyloid plaque formed at the synapse of neurons. Now, once amyloid plaque forms, it triggers an intracellular process that erodes all the nerve cell synapses with other nerve cells, causing memory loss, damaged cognitive abilities, and motor coordinations.
The current treatments for Alzheimer's disease focus on decreasing its symptoms. The two classes of medications to treat Alzheimer's are cholinesterase inhibitors, or CHEIs, and N-methyl D-aspartate, or NMDA, receptor antagonists. Both types of medication work by slowing down the process of cognitive degeneration after the plaque has already formed, by increasing the amount of available neurotransmitters, which are essential in nerve signaling. However, experiments showing the efficacy of these medications have conflicting results. Although they made some small but significant improvements in slowing the rate of cognitive and functional decline, they are not a complete cure. The research paper they were about to introduce is written by Kim T and colleagues at Stanford University, which was chosen as the focus of our presentation because it specifically identifies one of the possible receptors that beta amyloid binds to in order to create plaque on the neuron synapse. This paper suggests a different approach to understanding the cause of Alzheimer's after discovering a new element in the beta amyloid pathway, a murine membrane receptor PIRB, or paired immunoglobulin-like receptor B. We're going to divide the explanation of the research paper into three major parts, including the results and a discussion for each part. The first part is about proving that PIRB is the A-beta oligomer receptor, the second part is about finding the human version of PIRB, and the third part is about assessing whether or not PIRB contributes to cognitive deficits in a living mouse.
While downstream signaling pathways of A-beta are well understood, the upstream signaling pathways are not. However, in a recent experiment, researchers found that APP mutant mice that lack PIRB receptor have normal visual neural activity unlike other APP mutants. So, the researchers speculated that it is possible that A-beta acts through PIRB receptors. To test that PIRB is a receptor of A-beta, they prepared cells that express mouse PIRB and measure the binding of A-beta to the cells. What you're looking at in figure 1 is the A-beta and PIRB that are cell-stained. If you look closely at the arrowheads, you'll notice that there is a colocalization, which are the spatial overlaps between the red, the A-beta, and the green, PIRB. This proves that there is a relationship between them. What's more interesting is that A-beta shows a strong association with PIRB only when it's an oligomer and not when it's a monomer, as shown here in figure 2. Remember from earlier that A-beta tends to bunch up and then bind strongly to the receptor? Well, the fact that A-beta oligomers have a strong affinity only when it's an oligomer confirms that A-beta acts through the PIRB receptor. Similar experiments were conducted to compare oligomeric A-beta's affinity to PIRB with its related receptors, PIRA1, PIRA4, and isoform PIRB. Figure 3 shows that PIRA1, PIRA4, and isoform PIRB all have a much lower affinity than PIRB. This finding indicates that A-beta 42 oligomers bind selectively to PIRB.
While PIRB, paired immunoglobulin-like receptor B, acts as a receptor for oligomeric A-beta in mice, the human homologue of PIRB is leukocyte immunoglobulin-like receptor B. There are 5 family members in this receptor, leukocyte B1 to leukocyte B5. To find which of these receptors functions analogously to PIRB as a receptor for A-beta oligomers, leukocyte B1, 2, and 3 were examined because they are most related. Figure 4 looks at the amino acid sequence homologies to PIRB to leukocyte B for preliminary comparison. Leukocyte B1, B2, and B3 were 47, 48, and 53% respectively similar to PIRB amino acid sequence. Even though those 3 receptors are similar, Figure 5 shows that A-beta oligomer binds selectively to leukocyte B2. Also, leukocyte B2 proteins were detected in human brain from both Alzheimer's disease patients and non-AD adults, with no significant difference in levels. This suggests that leukocyte B2 may serve as a receptor for A-beta oligomers in human brain. In PIRB and leukocyte B2, A-beta oligomers appear to bind more to a particular N-terminal Ig domain, D1-D2. As seen from Figure 6, A-beta 42 oligomers fail to bind to the receptors PIRB and leukocyte B2. These results show that D1-D2 domains mediate binding of A-beta 42 oligomers, meaning PIRB and leukocyte B2 are possible receptors for said oligomers. In short, it was identified that leukocyte B2 in human brain as A-beta receptor that may lead to synaptic loss in Alzheimer's disease.
To test whether PIRB contributes in vivo to cognitive deficits, a mouse with the Alzheimer's gene and a mouse that lacks PIRB were crossed to produce a hybrid mouse with the Alzheimer's gene that lacks PIRB. First, recognition memory was examined using 2 tests, novel object recognition and novel place recognition. While the Alzheimer's mouse with PIRB showed impaired behaviors in both tests, the hybrid mouse didn't show any learning and memory defects. The hybrid mouse was also immune to synaptic loss. This result turns out to be because of the inactivity of an enzyme called coffulin. Coffulin breaks down actin, and no actin means no synapse, because actin is essential to maintain synaptic structure. In the Alzheimer's mouse, A-beta PIRB binding recruits coffulin signaling modules to facilitate actin depolymerization, resulting in synaptic loss. This connection between coffulin and PIRB supports the hypothesis that PIRB regulates synaptic plasticity in the Alzheimer's model. Soluble A-beta oligomer fragments acting as a decoy that selectively block leukocyte B2 function may be promising for treatment of the Alzheimer's disease, even in the prodromal stage.